So this is how filtration reabsorption work. Here I've drawn up the proximal convoluted tubule. Right here we have like the glomerulus, then the start of the nephron right here. Basically what will happen is you'll get filtration first through the fenestrated capillary, then through the basement membrane, then through they're called podocytes, which separate by size and charge, and then through mesangial cells. And the things that'll make it past have a size of around 6 nanometers max, and then they don't have any charge. And so we filter out a lot of things, but we want to reabsorb a certain amount of things. So what we want to reabsorb mostly is glucose, lactate, amino acids, sodium ion, chloride, calcium, potassium, bicarbonate. Important for buffering. These are very important, and we don't want to lose these in high amounts in the urine. Urea, which we'll talk about later, is used in this process that cycles so that we can highly concentrate the loop of Henle in juxtamedullary nephrons. We also want to get rid of metabolites. Alpha-ketoglutarate, which we'll go over later, serves as an anion metabolite exchanger. So it's how we get rid of some of these metabolites, is like toxins built up from metabolism. Okay, so first we're going to go over this proximal convoluted tubule, which is located right here. These are the cells that will pick up the filtrate here. And what happens is we have these sodium symporters that will bring in these filtrates that we want to keep. Glucose, lactate, amino acids. Those get filtered 100%. Now salts, we talked about how sodium, there's a high sodium concentration because on the basolateral side there's these sodium potassium pumps pumping out sodium here. There's a high amount of sodium right here in the interstitial space. We remember water follows sodium. Water will follow the sodium, and as it follows sodium, it also brings with it dissolved calcium and potassium, which diffuse back into these capillaries via the blood. These sodium symporters bring in glucose, lactate, amino acids 100%. I'm going to draw this right here, 100%. It's very efficient. And also we have a sodium proton exchanger, which is good for maintaining pH. We also have a sodium bicarbonate symporter, because in here in the cell there's a carbonic anhydrase. I'll draw this in another cell down here. Carbonic anhydrase, which converts CO2 and water into bicarbonate and protons. Protons will get transported via an antiporter out into the lumen, while bicarbonate via symporter will be transported back into the blood. Another thing to note about these transporters is that they can become saturated. If they do become saturated, then we'll have some of these filtrates end up in the urine, like glucose, lactate, and amino acids. Which shows clinically, it's important. It shows that maybe we have too much glucose in our blood, and that would mean diabetes. And so this is the proximal convoluted tubule. It does most of the work as far as reabsorption goes, but there's a lot more left, and we'll go over it. Here I've drawn up the proximal straight tubule. This is where it's located on the nephron. As the nephron comes down, we have the proximal straight tubule. And the main role of this one is to get chloride back into the bloodstream. So we have this CFTR, cystic fibrosis transport of chloride channel, transports chloride, potassium leak channels, and we have aquaporins getting water back into the bloodstream. I mean, as I said before, osmolarity will stay constant, and then we have this this sodium proton antiporter and this chloride anion antiporter to get these organic acids that we might produce in metabolism out of our bodies. Also, as you can see, proton and acid can come together and they can form that. They can diffuse it in the membrane. So this is how we get about 35% of our chloride back into our bloodstream. Now I'm going to talk about the descending and ascending tubule. Descending tubule is pretty easy. We have this sodium, potassium pump right here. Now it has aquaporins all throughout the membrane right here. And water will diffuse. It will follow the sodium right into the blood. And because all this water is going back into the blood, what it will do is it'll concentrate the urine down to 600 milliosmolar if we're talking about a cortical nephron here. The descending tubule is located right around this loop of Henle here. We have it here. This is the descending tubule right here. Ascending tubule will be right here. So, ascending tubule. This is where salt exchange occurs. We have this sodium-potassium 2-chloride transporter. It's impenetrable to water, so we're done reabsorbing water. We want to keep the amount of water that we have in to be urinated out. And so what will happen is salt will start to leave the solution right here, and it will dilute the urine down to 100 milliosmolar. Now this will be important later. This is an important control step because diuretics will block this, preventing so more water will stay in the urine. In the distal collecting tubule, you can see that it is tightly regulated by ADH and aldosterone. Aldosterone will increase sodium potassium pumps, and ADH will cause the release of aquaporins. Aquaporins go to the membrane and allow water to go through, decreasing urination and increasing the amount of water in the blood. This is an important regulatory step. Basically, the body can control how much water it wants to let go in the urine or how much water it wants to maintain. Not a whole lot much more to say. Most of the sodium, potassium, calcium, chloride, other important minerals, electrolytes are conserved, but very few will get excreted, such as potassium. All right, let's talk about filtration. So the glomerulus here has a very high hydrostatic pressure compared to other capillaries. It's 55 compared to 30, which is the normal hydrostatic pressure for other capillaries. It's extremely large, and this is in order to filter things out, and it's to overcome the capsule pressure of the tubule here, which is pushing back, and so BCOP is also pushing back. Means a total pressure of 38 going into the capillary, but we want things to come out. So that's why we have such a high hydrostatic pressure. And there's 4 layers that things must get through in order to make it into the capsule. And here, the first layer is the fenestrated capillary. There's little slits in the capillary, kind of like this. And these separate things by size. The basement membrane, size and charge, it's negatively charged. And so that will prevent most things from coming in that have a charge. And it also separates them by size. Podocytes, which size and charge are like these little cells with these little fingers that have negative charges on them, and they will also separate things by size and charge. And then the 4th layer are mesangial cells, which is like smooth muscle. And so things that get through are uncharged, and they're about 5 nanometers in size. So that is how things filter into the capsule. So the total rate at which things filter into the capsule is called glomerular filtration rate. And this, simply put, is just or I guess clearance. This is glomerular filtration rate, how fast things just get into the Bowman's capsule. Whereas clearance is, if I draw out a nephron here, clearance is how fast things, or like the rate at which things get into the collecting tubule here. Here's another nephron. So how do we figure out clearance? Well, clearance, oops. All right, clearance equals GFR plus secretion minus reabsorption. I'm just going to call it Re-A. So to find our clearance, how fast, the rate at which something gets in, because right, we have filtration, reabsorption, secretion, excretion, right? And so we need to figure this out, but we can't just like take glucose, because the GFR for glucose is going to be different than the clearance for glucose. We won't have any clearance of glucose unless you have diabetes. So here's what we do. For clearance to equal GFR, a substance must be freely, almost filtered. It must not be secreted. Secreted. Okay, I'll be out there in a sec. Or not be reabsorbed. That is how clearance equals glomerular filtration rate. And what they did is they found something called inulin. Now, inulin is not made by the body, not used for anything, and so it's freely filtered. It's not secreted or not reabsorbed, so it just gets excreted freely. And what they found something very similar to inulin called creatinine. And that's why we run blood creatinine just to get a measure of your clearance. So that is clearance. Okay, right now we're going to talk about glomerular feedback. Let me put this in. Oh shoot, okay, maybe I should. Okay. Okay, so our kidneys are very sensitive to changes in pressure, because changes in pressure can alter the changes in filtration rate. So for example here, we have an increase in hydrostatic pressure that causes an increase in glomerular filtration rate. And now, how do we regulate that? Well, we have this thing called tubuloglomerular feedback. There are sensors in the macula densa that are like, yo, we're filtering out way too much crap right now. So like, they're going to release adenosine, and this will cause vasoconstriction in the efferent arteriole, causing a decrease in GFR, which is what we need. Okay. Say we have a decrease in hydrostatic pressure. We will have a decrease in GFR. Thus, the macula densa would be like, I'm going to call MD, be like, yo, let's release nitrous oxide, which is what's in pre-workout for vasodilation. And this causes an increase of GFR. Vasodilation of the efferent arteriole will cause an increase in glomerular filtration rate. Okay, the Ross system sort of works like this. This is another way that the kidneys regulate feedback and to increase glomerular filtration, right? We have a decreased GFR, usually like that due to decreased blood pressure. Thus, the kidneys will start releasing renin. Renin then converts angiotensinogen into angiotensin 1, which then through ACE1 gets converted to angiotensinogen 2, and then angiotensinogen 2. ACE2 converts that to angiotensin 2. This is a potent vasoconstrictor, increasing blood pressure. Goes to the adrenal gland, producing aldosterone. It's hormone which perfuses into the cells, increasing the transcription of certain genes that affect blood pressure. Goes to the brain, triggering thirst and releasing ADH, which means we will pee less, which means we get rid of less body water, increasing our blood pressure. Increased volume, increased blood pressure. Baroreceptors increase ADH as well due to low stretch. And this is how this system works. It's very important that we have it in place. There's another side to it called angiotensin 1,7, which decreases blood pressure. It's how this is regulated. So that's how it works in a nutshell. Okay. This is another little diagram of the nephron, but folded out. Kind of looks like a snake. What? Nephron. I said it right. It's nephron. You said nephron. No, I said nephron. Okay, whatever. Continue. I said nephron.
I'm his sister. Bye.
Okay, so the proximal convoluted tubules right here. Got the proximal standing tubule. Goes down right here. We have the loop of Henle. Then here is our ascending tubule and our descending tubule. This part's important for making the urine a little bit more concentrated. It's permeable to water here, so water will flow out. This will make the urine more concentrated as the concentrates stay behind. This part can be regulated so that we can either have more or less water coming in. Right here, sodium and chloride, we have these like salt channels here that will leak out and leave behind the water, making it more dilute solution. And in the collecting duct here, basically it can be regulated. How dilute do we want our urine? And that's why we got to go to urea. Urea is very important. Here, this is important too, as ADH inhibits. So ADH blocks these salt channels, basically preventing salt from leaving. Thus, water won't follow. Or they prevent salt from coming in, I should say, sorry. They prevent salt from coming in. Thus, when water follows, it won't follow as much, and the urine will become more dilute. Thus, with ADH, the osmolarity of our urine is like 1,000. Without it, it's around 100. So, and getting a urine of that high osmolarity, usually done over here in the juxtamedullary nephron. ADH will make urea more permeable. And so why in the juxtamedullary neuron here in the descending portion of the tubule, the fact that the urea can cycle makes it very concentrated, as water will follow it out, leaving behind this urea. And regulating this will make it so that our body controls how much water we have in it. It's a very good way of controlling blood pressure.